Welcome, everyone. I'll start our proceedings today. This is the webinar on protein engineering for biotechnology and enzyme catalysis, which is brought to you by the Protein Society. My name is Liz Meyering. I'm the president of the Protein Society, and I'm really delighted to be introducing our first webinar of 2024 on the topic of protein engineering for practical purposes, which is going to be given by leaders in this exciting field from both academia and industry. So you have the graphic here. Before I go to the details of the science, I'm going to start with just a few general announcements and point out that, as you can see here, this webinar is part of a series that the Protein Society has been hosting. Our next webinar is on February 21st, which will be on navigating the structural frontier with protein NMR in the era of artificial intelligence. So we look forward to that next webinar, and we invite you all to make suggestions for topics and to consider organizing a webinar yourself. We have wonderful staff at the Society who make this really easy to do. So you can contact me or Raluca Kedar, our CEO, who our information is on the Protein Society webinar webpage. And I'd also like to thank Raluca and Shannon Univ, our communications manager, for all their help behind the scenes in making this webinar possible. So my first announcement is an invitation to join us at our annual symposium this year, which will be in beautiful Vancouver. It's taking place in July. And our registration is open. We have a fantastic program, and you can find all the information for the symposium at the Protein Society website. Oops. I also wanted to highlight our journal, the flagship journal for the Society, Protein Science, which is led by our editor, John Kurian, and his editorial team. This journal is flourishing, and I really encourage you to consider publishing your own exciting research on proteins in this journal. And with that, I would like to turn to the main focus of our webinar today. So I'd like to start by expressing our warm thanks to our speakers, Uwe for envisioning and leading this webinar, and Florian and Ingmar for joining us to tell us about your amazing work at the forefront of this tremendously important field of applications of proteins and enzymes. So the structure of our webinar is that Uwe is going to first give an introduction to the topic of the webinar, followed by his talk, and then we'll have talks by Florian and then Ingmar. And I'll first now just give a brief introduction to our speakers before turning it over to Uwe. So Uwe Bornscheuer's work throughout his career has been at the forefront of multiple waves, as he calls them, of protein engineering for biotechnology and enzyme catalysis. His PhD was from the University of Hanover in the Department of Technical Chemistry with Professors Schuster and Schepper. He did a postdoc at the University of Nagoya in Japan with Professor Yamane, habilitation at the Institute for Technical Biochemistry at the University of Stuttgart with Professor R.D. Schmidt. And since then, he's been a professor at the Department of Technical Chemistry and Biochemistry at the University of Greifswald. His work on enzyme engineering and lipid technology has been recognized with many different awards from Germany, from Europe, from the United States, from Korea, Thailand, and Japan. And he serves in editorial roles for many journals in these fields, as well as advises to companies. Florian Hollfelder worked with Dan Hirschlag at Stanford, Tony Kirby and Danny Tufik at Cambridge as a student, then as a postdoc with Chris Walsh at Harvard Medical School, before returning to Cambridge to establish his own research group in the Department of Biochemistry, where he's a professor in chemical and synthetic biology. So his research program centers on quantitative and mechanistic questions at the interface of chemistry and biology, using low and high throughput approaches that we'll hear about today. And he leads multiple EU-funded transnational collaborative initiatives that are centered on proteins. Ingmar Schuster did his PhD in computer science, so comes from a different direction, at Leipzig University with Gerhard Heyer, then postdoctoral research at Université Paris-Dauphine and Freie Universität Berlin with Christian Robert and Christoph Schutte, and further developed his expertise then in machine learning approaches. He has worked in various companies and is co-founder and CEO at Exazyme, a company that has developed an AI protein design tool that helps biotech innovators more efficiently develop proteins for applications in diverse areas, from food production to biotech to medicine, and, to quote from their site, to overcome humanity's greatest challenges for humanity and beyond. So we're really excited to have these different perspectives brought by our speakers today. I'm really looking forward to this. I'll give you just a couple of logistical notes. So the way that we're going to do this is that each speaker will give their presentation, and we'll have questions after each presentation, just a short question period of about 5 minutes. After all 3 talks, we'll have a more extended question and discussion period. So you can post your questions at any time using the Q&A. And so you can look for that tab in your Zoom meeting. When you type your questions, they will only be visible to us who are on the panel here. They will not be visible to the general audience. And then I or Uwe or the speakers will read out the questions in the question periods. We likely won't be able to get to all the questions in real time, but you can also see answers to questions which the speakers may give in the chat, so they'll be typed. And after questions are answered, they will be moved so that everyone can see them. So with that, I'd like to also thank all of you in the audience for being here. Your participation is essential to our discussion and moving us forward. And with that, I'll turn it over to Uwe.
Yeah, thanks a lot for the kind introduction. Just a second, I want to get started. Can you see the presentation, everybody? Okay, so first of all, thanks a lot, Elizabeth, for making this possible, for approaching me and for asking me to organize this webinar. And thanks, of course, to your team for being so professional in getting everything organized. And of course, special thanks to Florian and Ingmar, who also agreed when I asked them to give a talk, because I think the science they do is perfectly complementary to all the things I would like to present now. So I just have to find the mouse. Yeah. That's the wrong one.
Yep, I see your mouse.
You see it, okay? Yeah. And I just have to delete this here. Okay, so because, just a second, now it doesn't go back.
It does.
So yeah, here we are at the start. So protein engineering is, of course, a very wide field, and the Protein Society has so many members from various disciplines, where the binding interest is, of course, anything about proteins, but not everybody uses proteins for applications in biocatalysis and biotechnology. And therefore, I also would like to give everybody an introduction into our motivation, into our discipline, and why protein engineering is so important for us. Briefly, some info is about Greifswald, where I'm located. So we're in the northeast corner of Germany. Somewhere here is Berlin, somewhere here is Frankfurt. So we are right at the Baltic Sea, which is a nice place. Even in winter, it's very touristic here with very beautiful islands close by. We have a nice fishing, yachting harbor, city center with a cathedral where the university was founded in 1456. So it's one of the oldest German universities, but also the oldest Swedish university, because when they had occupied this area, they had no university yet in Sweden. We have about 10,000 students, 5 faculties, and a very strong focus on natural and life sciences. This is our building of the Institute of Biochemistry, where I'm located. And this is the aula in the main building of the university for official things. We, of course, meet there. So I will give you first an introduction, as I said before, to tell you why is enzyme engineering important for biocatalysis. And then I will show you 2 recent examples from my research about the design of amine transaminases, but also for P450 monooxygenase. And then I will close with an outlook and conclusions. So why do we do biocatalysis? I'm a chemist by training, as you have learned. And of course, there are many chemical synthesis methods established since centuries. and everybody wants to convert a substrate into a product, whether you want to develop an enzyme to convert, to break down plastic for recycling, or whether you want to make a carbon compound. We have a reaction which we want to have catalyzed by an enzyme. So we have to identify the right enzyme. That can be classical by screening in nature. You can screen in strain collections and identify maybe a certain microorganism which has the enzyme of interest. You can use isolated enzymes, and if you are lucky, they are commercially available and you don't have to do any enzyme engineering. Always we have to do the characterization of the biocatalyst. That means we have to determine the kinetics, kcat over km, for instance, the pH and temperature profiles, the substrate scope, the stereoselectivity, and so on, in order to know whether the enzyme is really suitable for a given process. And very often we end up here, we need to optimize the enzyme. And that can be explained, for instance, by the fact that a lipase, an enzyme which hydrolyzes fats and oils, triglycerides, is designed in nature for exactly that specific reaction. So it chops off fatty acids from the triglyceride at low substrate concentration and maybe at body temperature. And then we want to use the enzyme for completely different things, synthesis of optically pure alcohols, for instance. We want to work at 100 grams per liter substrate concentration and maybe in 10% organic solvent. And the enzyme is not designed by nature to withstand these rather harsh conditions. And then we do enzyme engineering by directed evolution or rational design, you will see in a minute. And if we are successful, then we have a good catalyst for the reaction of interest. And then the real engineers have to think about, how do we run the large-scale process? Do we need to immobilize the enzyme? How do we do the reaction itself? How do we do the downstream processing for the product isolation? And if everything works out, at the end, there is a new biocatalytic process, which is maybe competitive to a chemical process. So why biocatalysis? Of course, we all know that enzymes catalyze a very broad range of chemical reactions. They can be very efficient. What's also good is that they work under mild reaction conditions, especially when you compare this with many chemical processes where you either work at minus 78 degrees centigrade or at maybe 150 degrees centigrade. And with enzymes, we usually work somewhere between room temperature and maybe 60 degrees or so. That saves a lot of energy for the process, but it also enables you to convert sensitive compounds easier because you can avoid side reactions. Isolation of the product is often very easy, especially if the enzyme is immobilized. You can just filter it off. And most important is that enzymes have the feature of chemo, regio, and stereoselectivity. That means you can make a direct synthesis to the desired product, the optically pure product, in one step. It means you have no or less byproducts. There is no need for protecting group chemistry, and usually no need for toxic reagents, no transition metals, no toxic solvents or other things. And of course, synthesis of optically pure compounds is possible. For instance, for limonene, the two enantiomers have totally different properties. This smells like orange, this smells like lemon. This you can also find documented in textbooks, which I have written together with colleagues. And just 2 years ago, we published a big review in Angewandte Chemie about the state of the art for the industrial application of enzymes. And why do we do the protein or enzyme engineering? This can be very basic science. We want to understand how an enzyme functions. And that means not only identifying the residues involved in the catalytic reaction. It can be even more. What makes an enzyme a dimer and not a monomer or something like that? We want to understand evolutionary relationships. What have been the ancestral enzymes of a modern enzymes? Why do ancestral enzymes maybe can catalyze two different types of reactions, but the modern enzymes are very specific enzymes, and this is also basic research. But of course, you want to alter the properties of the enzymes for a given application, as mentioned before. Maybe the target is to change the substrate range, which can be a broader substrate range. So they have one enzyme to convert 100 different related chemical compounds, 100 different alcohols, 100 different ketones. Maybe you want to make the enzyme more specific so that the glucose oxidase is only oxidizing glucose, but no other sugars. Maybe you have to improve the stereoselectivity because a wild-type enzyme only gives 80% optical purity, or you found a very good R-selective enzyme and you want to make it S-selective, and then you can invert the enantiomer preference. Often we have to reduce inhibition, which especially occurs if you work at high substrate or product concentrations. Often the enzyme needs to be made more tolerant to organic solvents, which we need to add to solubilize the substrate. Sometimes it can be the temperature range, the temperature stability, the pH range, and in a few cases, just improved expression can be a target of an enzyme engineering experiment. That means if you have a wild-type enzyme with certain properties, shown here in a bluish color, and you want, then you often have to improve this. For instance, if you want to make it more selective, then you can target enantioselectivity or regioselectivity. At the same time, you want to have the improved enzyme to be maybe faster. Expression should be good. It should be stable. Also for storage, should be stable against heat, solvent, reactive components to avoid inhibition, or to adapt the enzyme if you want to couple 2 or 3 enzymes in a multi-step pathway. And it can also be that you have an esterase with very low amidase activity and you want to make the amidase activity more pronounced. Then you can try to shift this minor activity to a major one, or you repurpose the enzyme to catalyze completely new chemistry. What you have to keep in mind if you introduce mutations which improve selectivity, it can easily happen that the enzyme is more selective but less stable. So in the ideal world, you would try to optimize the enzyme for various properties at the same time. Of course, enzyme and protein engineering is a very wide field. You can use rational protein design, which I will explain in a minute. You can use directed evolution or combinations thereof. If you do directed evolution, you need high throughput tools, microtiter plate assays, microfluidics, as you will learn soon from Florian Hollfelder, maybe growth assays. You can look at catalytic promiscuity as an evolutionary concept. You might have to work on display methods to screen very large mutant libraries by phage display or auto display. You might consider cell-free protein synthesis. You wish to incorporate non-canonical amino acids into proteins to catalyze completely new chemistry. You can design the proteins on the computer. Even de novo computational design has been described. You might consider the effect of post-translational modifications like lysine acylations to change the properties of the enzymes. And of course, we also have to have people to do protein crystallization and X-ray structure determinations, even in the times of AlphaFold. And of course, the target proteins can be enzymes, antibodies, transport proteins, binding proteins, etc. And I guess many of you know that in 2018, the Nobel Prize in Chemistry was given for protein engineering to these 3 winners, Francis Arnold for the directed evolution of enzymes, and George Smith and Sir Gregory Winter for developing the phage display method of peptides and antibodies. So what strategies can you choose from? And this picture is a little bit older, but it's still valid, because on one hand, we have the well-established rational design. Someone determines the structure of the protein. Maybe you only have a homology model, or nowadays maybe an AlphaFold model. Then you dock the substrate into the active site using your computational methods. If you are lucky, there's even an X-ray structure in high resolution which contains a transition state analog. Then you know more exactly how a substrate binds to the active site and how the binding pockets look like. And then you try to predict which mutations will, for instance, enable the enzyme to accept a more bulkier substrate. All of this works, and usually the prediction based on rational design requires the creation of a handful of mutants and not millions, but it's very information intensive. If you don't have the X-ray structure in high resolution without transition state analog and just a lousy homology model, don't be surprised that your prediction is not so good. That's why everybody was happy in the early 90s when the directed evolution idea was born, which in principle means you take the gene of interest, you do error-prone PCR to introduce randomly mutations, or you do gene shuffling, also called DNA shuffling, invented by Pim Stemmer. You can do insertion deletion mutations and other methods. And then the challenge is the screening effort, because you easily end up with mutant libraries containing millions of variants or even more. And in the last 2 decades, people combined these approaches. You have a homology model or a structure of the protein, you know where the music plays, but you don't know exactly which amino acids at these 6 positions you should exchange, and you also don't know which would be the best substitution at each of these positions. And if you consider 3 positions in the active site, you have 20 times 20 times 20 possible combinations. That makes 8,000. Keeping in mind the codon bias, you should screen roughly 100,000 variants for only 3 positions in the protein. That can be just 1% in a 300 amino acid protein. So you cover only a fraction of the sequence space, but you still need to analyze 100,000 variants. And this is also shown in this table. The challenge is often how to find the needle in the haystack. Of course, Shaun the sheep is very bright and finds the needle immediately. We have to have sophisticated assays. To give you an idea how big the sequence space is, if you have a protein with 200 amino acids in length, you introduce 3 substitutions randomly, there are 9 billion possible combinations. So that is 1.5% of the sequence space. So you need a rapid, to save time, and a reliable assay system to run these directed evolution experiments successfully. And the throughput varies considerably depending on the specific problem, the assays you have at hand, and that is where we usually spend most of the time in a protein engineering effort to develop a suitable assay. So in general, my group, but also all others, I think, use this concept. You have a structure or model of your protein. You dock your substrate to the active site. If the property is located, can be assigned to the active site. If it's thermostability, maybe you have to exchange residues on the surface of the protein. But if it's a stereoselectivity, clearly you have to introduce mutations around the active site. Then commonly we do bioinformatic analysis, and machine learning is part of this nowadays to figure out which mutations at which positions shall you introduce at best. So you predict this, and then someone indeed has to go to the lab. Even if you order synthetic genes, someone has to put this into E. coli and then put this on an agar plate, and then you pick the clones. You might analyze them in microtiter plates, so you have to grow them, express the protein of interest, lyse the cells, analyze this, and if you find a hit in your microtiter plate, of course, you must verify this by classical analytics like GC or HPLC. To give you an example for this, some years ago, we looked into engineering an esterase. We had a alignment of the alpha-beta hydrolase fold superfamily called 3DM database. It contained in this specific analysis 2,813 enzymes, which includes, of course, lipases, esterases, epoxide hydrolases, dehalogenases. And then we looked into this database to figure out at which position shall we introduce which mutation. And we didn't do saturation mutagenesis. We looked carefully which amino acid substitutions occur frequently within thousands of natural proteins. We called them allowed substitutions, and those which rarely occurred, we called not allowed amino acid substitutions. So we could make the library much smaller and much smarter. And the student made the library. We looked for thermostability, and in another paper also for stereoselectivity. And this is a library which contains all forbidden amino acid substitutions. Most variants are neither thermostable nor active. If you do a normal NNK saturation mutagenesis, of course, you cover all the members of substitutions at these positions. You see there are hits also in this corner. And in the allowed library, the designed library, we have even more hits. And the good news is, or worse for the student, here we found about 25 hits in 1,000 variants. Here we found about 5 hits. So you can also save a lot of screening effort in the best variant with 8° more thermostable. To facilitate the screening, we have a robotic platform which allows you, beside colony picking into microtiter plates, to do all the required steps in an automatic fashion. So E. coli are grown in the microtiter plate, you induce expression, then you lyse the cells, you can do centrifugation in the centrifuge, you take the supernatant, and then you do your biochemical assay in 2 microtiter plate readers, so you can measure, let's say, activity and stereoselectivity, and also determine protein content if you wish, and get a specific activity. At the end, you need, of course, sophisticated data analysis, and you can easily screen about 20,000 clones during a week with all the requested kinetic data. If you want to go for even higher throughput, we have a microfluidics platform, but Florian Hollfelder has even more sophisticated systems, and he will tell you very soon how he uses microfluidics to identify new enzymes. About 11, 12 years ago, we published this review in Nature, where we said at that time we were in the 3rd wave of biocatalysis. What does that mean? 100 years ago, chemists or biochemists could already use enzymes to make organic synthesis molecules like this hydroxynitrile. In that case, the enzyme came from bitter almond extract, and it coupled benzaldehyde and HCN in a stereoselective fashion. The problem was, if you have to make 100 kilograms of this compound, you need a lot of bitter almonds. So supply of the enzyme was a problem. And also, you couldn't do any protein engineering. In the 80s, gene technology was invented, so you could clone a gene and express the enzyme recombinantly. And then, of course, you could scale this up because you can produce a lot of recombinant enzyme in a big fermenter. And then also, people started to use rational design, and as I mentioned before, directed evolution with error-prone PCR was studied. So you combine random mutagenesis with high throughput screening, and the challenge was people had to screen very large libraries. At the turn of the century, we thought that this is now the 3rd wave, because we now can order recombinant synthetic genes. So we save all the time on cloning, but at the same time, we can also introduce already in the synthetic genes the mutations we want. So you can order a library of variants containing the desired mutations. Based on the computational analysis and all the information in protein and sequence and structure databases, we can make projects faster successfully, and we can run them successfully faster. And at the same time, the chance for success has increased tremendously. To give you one example, we designed amine transaminases to convert bulky ketones. And we started with compounds like this. We worked a lot on transaminases in the past. And what these enzymes are doing, they take a ketone and convert it into the corresponding amines. That's why they are called transaminases. Of course, they need an amine donor. This can be, for instance, alanine, and this is converted into pyruvate. And the benchmark substrate for, I think, all transaminases is acetophenone converted into alpha-methylbenzylamine, also called alpha-phenethylamine. And the enzymes show good activity. In this case, for this enzyme, for is about 1 unit per milligram. 1 unit is micromoles per minute, and that is a good activity. But if you change the methyl group to the ethyl group, you see activity drops by a factor of 10. If the methyl group is turned into a hydroxyl group, activity is more or less gone. So this is not even what a chemist would call a bulky ketone converted into an amine. It's a small change on this side of the carbonyl function. And that is because the enzyme has 2 pockets, one for the aromatic ring and a very small pocket on the other side. So we selected certain positions in the binding pocket of the enzyme based on a 3DM analysis, and also performed site saturation mutagenesis and screened them with a high throughput assay. in the microtiter plate where we measure acetophenone formation. So you go this direction in the assay because this you can easily measure. And then we combined the best hits. And then at the end, we could show we get complete conversion, perfect selectivity using alanine as the amine donor. And this is shown in the table. Activity improved also substantially for the normal substrate acetophenone. But here with this double mutant, suddenly you can convert this compound efficiently into the corresponding amine. And even for the hydroxyl substituted compound, we get reasonable activity with a double mutant. After publishing this, we got a new project, and these ketones are indeed very bulky. So again, the benchmark substrate acetophenone, and these compounds clearly require a large, very big second binding pocket in the protein. The second challenge is if there is a lot of space in both binding pockets to accommodate this ketone, it can flip around, and then you might get the amine, but you might get racemic product. And the same challenge is here, and also for this more symmetric compound. So we thought, how can we address this problem? And we didn't use directed evolution for these three compounds. We used rational design. We looked at the transaminases from 3 different organisms. These enzymes are well studied since more than a decade, and you learned before that they hardly convert ketones with a side chain bigger than methyl. Another enzyme from Regeria species, 3-FCR as a PDB code, shows very little activity towards the 2 amines. But it was also not very active towards the benchmark substrate. And we looked into the structures, and it was very difficult to see any differences in these 3 proteins, which might explain why this one has some activity, the others have not. Based on this analysis, we identified 2 substitutions, introduced them into 3-FCR, and you see from no activity for this compound, 0.001 for this compound, that is a very little activity mentioned before. We could increase this substantially to almost 1 unit, also substantially here, and a slight increase here, and a first sign of success for this challenging molecule. And then we performed even more sophisticated rational design with molecular docking, energy minimization of the quinoid intermediate, identified more positions, and introduced them stepwise. And at the end, we had a quintuple mutant, which gave also good activity on this compound, very high activity on this one, and very good activity on these 2 molecules. So by rational design, we could substantially improve activity. A side note, this variant is the most active one, but this is also the most unstable one. So for preparative purposes, we had to remove this proline histidine mutation because the enzyme simply was not stable enough. And then finally, we could show we take these compounds shown on the previous slide, the very bulky ketones, we get complete conversion, we get good isolated yields on preparative scale, we got excellent selectivity for compound 2, we got a very good optical purity for compound 1, and compound 3, just to go back, is this very symmetrical compound where it's even more challenging to get stereoselectivity introduced. And all these variants also converted the substrates shown before with very good activity and selectivity. Just recently, and for time reasons I cannot show this, we published a fine-tuned growth selection system to find better transaminases, monoamine oxidases, and phenylalanine liases. We created mutant libraries in the range of 10 to the power of 5 variants for each of the enzymes for a given synthetic problem, and performed ultra-high throughput screening in this sophisticated growth selection assay. And related to the talk by Ingmar Schuster, we also just published a paper where we used machine learning to guide the engineering of transaminases, which again was very successful. Now I would like to switch to another enzyme class and another challenge, engineering regioselectivity of a P450 monooxygenase. P450 monooxygenases catalyze hydroxylations, so they can convert a non-activated carbon into a corresponding alcohol. That is well known. And we had a specific target compound called UDCA, and this is ursodeoxycholic acid, and it's used to treat gallstones. The name ursodeoxycholic acid already implies it comes from this natural source, and I think it's not a good idea to ask this guy whether you can get the bile acids from him or her. So it's better to use another starting material and to find an enzyme which catalyzes the desired transformation. And the litocholic acid shown here is available on large scale and low price. And the challenge is, can we find an enzyme making hydroxylation only at this position, the 7-beta position? Of course, there are many other carbons in the steroid. We looked into numerous P450 enzymes and identified an enzyme called OLE-P as the best candidate. OLE-P was reported to be an epoxidase, but in fact it's a P450 monooxygenase. But this enzyme, the wild type, only makes 6 beta hydroxylation. So we had to teach the enzyme, forget about position 6, learn how to hydroxylate position 7. And of course, we also don't want to have a double hydroxylation in the molecule. So we looked into the structure of OLE-P. We used sequence similarity networks and also the 3DM software tool to identify possible hotspots and substitutions. We made then an alanine scanning on 24 positions, selected 5 positions to create small but smart libraries. And at the end, we screened roughly 4,500 variants. We also needed to develop a high throughput assay for this. And this is summarized on the slide. Here's the structure. Here is the active site region, and you can see the litocholic acid modeled into the active site, the 24 residues around the binding pocket. And then we performed all of this, and the wild type made no UDCA at all. And stepwise, we introduced mutations, and we found the triple mutant, which has perfect selectivity. We see only formation of UDCA and no formation of the undesired MCDA. And this was, of course, confirmed by HPLC and NMR spectroscopy. So we could show that it's possible to teach the P450 enzyme to hydroxylate another position and to forget about the 6-beta hydroxylation within this project. My outlook and conclusion. There are, of course, many trends in biocatalysis, and it's difficult to summarize them all on one slide. But what I think is really important is that we have better and better tools to identify suitable novel enzymes, but also to guide enzyme engineering. This spans from phylogenetic analysis over sequence similarity networks to computational tools like 3DM, KVR, Hotspot Wizard, Enzyme Miner, PROS, and many others. Of course, machine learning, not only PROSA developed by Codexis and published 2007, but many other new tools have emerged in the past few years. We will have the lecture by Exazymes in a few minutes. The computational in silico, de novo design of proteins, novel enzymes catalyzing new to nature chemistry, artificial metalloenzymes, the combination of photo and enzyme catalysis, and also enzyme catalysis with other disciplines from chemistry. And if you have multi-step synthesis in mind, you can also now plan a retrosynthesis for biocatalytic routes and can predict the best route for a given application. We have just published late November a new review, an update of the 2012 review, where we also summarize the most recent trends and developments for screening nitrous diversity. The sequence space has increased enormously. There are more than 2 billion sequences now available in public databases, so you have many proteins to choose from. For directed evolution, we have seen a tremendous improvement in the models and predictions, and you can easily, not easily, but it's not so challenging anymore to convert the enzyme, to teach the enzyme to convert not only the native substrate, but non-native substrates. Redesigning enzyme mechanisms allows you to do completely new chemistry in a well-known protein scaffold, and the computational design, of course, helps us also immensely to reach our goals faster and to make enzymes more successful. This picture shows the changes in the tools we have available and the developments, experimental tools. Enzyme repurposing is like using them for new type of reactions. DNA sequencing, of course, is cheap since a long time. DNA synthesis, there are new developments to make also DNA based on enzymes quite efficiently. High-throughput screening tools, automation is for sure very important and still needs to be developed further. Incorporation of non-canonical amino acids opens up new chemistry. Structure prediction with AlphaFold is now straightforward. What we still miss a bit is property prediction. Can we really predict whether a new protein we find, where we predict the structure, will convert a certain substrate at 100 grams per liter concentration within 5 hours and gives 99% EE of desired enantiomer? So we are not there to predict the property. Sequence mining is ongoing. Machine learning, extremely important. And then in the application, we see more and more cases for successful enzyme cascades, combination with chemical, catalytic methods for synthesis, upcycling, recycling of plastics, for instance, but also more applications in biologics and therapeutics. So I think I come to an end of my presentation to stop here now, and I should show this again. And then I think I'm ready for a few questions and answers. As mentioned before by Elizabeth, we will have a longer question and answer session after the talks from Florian Hollfelder and Ingmar Schuster.
Thank you so much. Yes, this was a great overview and telling us both what has been done and what will come. And there are a number of questions. Actually, they're rapidly accumulating here in the chat. So I'll just maybe paraphrase a few of them. We'll take a few now and then come back to them later, and you can answer them also in written form. So one thing that I think I see as a theme here, there's some questions from anonymous attendees and specific attendees, but a key question is, how do you decide what approach to take? So, you know, this is rational versus computational, different computational approaches. Is it very specific to your project?
Yeah, I think it's always specific to the project, but I would say it depends on a bunch of factors. I mean, if I have a high resolution structure of the protein, and of course I have someone skilled to deal with these computational programs, then I would, and the problem is related to something which I can address with mutations around the active site, I think I would always go for starting with a rational design. And see what comes out. Because you can start right now after this webinar, look at the protein structure, dock your substrate, and predict something. If the problem is too challenging and there is no related paper and literature where people have tried to solve a similar problem and you work on something completely new, then this, and there is no structure of the protein, then you do directed evolution. This is like 30 years ago when Francis Arnold and their team started to do this because they couldn't figure out why is this esterase not stable in the presence of an organic solvent. And everybody who can make a PCR can make an error-prone PCR. Then you need a good assay, and then that for that specific paper, it was relatively easy. It was a commercial compound, and after hydrolysis, it turns yellow. So you can start readily next week to screen your mutant library. And then the advantage is that you usually do not find a much improved variant when you do this random mutagenesis, but you find new hotspots. Suddenly, you see a 1.5-fold improvement with a mutation here in this corner of the protein. You never think about when you look at the protein structure. And then you do saturation mutagenesis there and might find an even better mutation. And I think what is best is keep your eyes open. Which strategy is the best? Even I cannot tell. We sometimes use different strategies or in parallel different strategies. I showed the example of the transaminase with the bulky ketones for another compound, which I didn't show for time reasons. We were stuck with rational enzyme protein design. We couldn't get further. And then we used arrow-prone PCR, had an agar plate assay to find the mutants. E. coli turn red if they are active. And then we could find a solution for the specific compound number 4, which was, even when we knew the best substitutions at the end, we would have never predicted this on the computer. So try to use all of the methods available, and then of course see, because the question was also, if we do not have enough money, what shall I do? Yeah, try to think about a simple and cheap solution. And the growth selection assay we just published last year is a prime example for a cheap solution, because you just need to have agars to make agar plates, and then you plate out the clones. And if they can grow, then you have a clone with an active variant. This is not expensive. No need for a high throughput robotic platform. Yeah.
Yes, yeah, no, you touched on many of the questions here. So maybe I'll just have one more quickly, which is related to that, which is obviously you have to compromise. This question comes from Michael Burns. In terms of what you can do. So often between the number of variants you can screen and the quality of the screen, for example, an assay as close to the desired process conditions as possible. So if you're going to have to choose between number of variants, given that you get these surprises, and the quality of your screen, how would you approach that? Which would you favor?
I would always go for an assay as close as possible to the desired process, because we have seen very often, hooray, in the lab. I found 10 mutants, which all are more active on the artificial surrogate substrate, and they all look better. And when we tested the real substrate under the real conditions, many of them were not better. And that is the first law of directed evolution, is you get what you screen for. If I have a big umbelliferyl side chain to get a good reporter molecule, then I should not be surprised that the evolved enzyme is perfect for the umbelliferyl modified substrate and is not so active on just a methyl ester of that compound, yeah? Because I screened for variants which can convert this compound. So if you can implement this in your assay, this is the best what you can do. We had an example 15 years ago where we had just an acetate, and we found the kit, commercial kit containing other enzymes to determine acetic acid. And then this kit worked perfectly, and then we had the real substrate and not a chromophore. So this is what one should do, yeah? I would say.
Okay. So with that, we'll come back to additional questions, and Uwe may type some in the chat. So I'd like to thank Uwe again and give the floor to Florian.
Okay.
Your screen. Thank you. Good. We see your slides.
Okay, perfect. Thanks, Uwe, for a great introduction to everything that's possible. And thanks also to the people who ask questions, because I want to start exactly at that edge of what is possible and what's not possible. So on one level, Uwe's talk has given us a wealth of practical methods that all work. He elaborated on them already. Shuffling, library synthesis, we can do that. We have 3D structures. We can predict some of them. We can predict mechanistically what might happen and what might not happen. And the kind of only thing that would maybe alert someone with knowledge in German literature would be that Uwe said it's a wide field. In Theodor Fontana's novel, Der Stechlin, there's an avuncular, friendly, grandfatherly figure with a lot of wisdom. And whenever he doesn't quite know the answer, he says, that's a wide field, because there is probably an answer, but it's not immediately obvious where it would be. And I want to focus today on things that are maybe not immediately obvious, where solutions are out there, but we need additional strategies in addition to kind of all these technologies, to the experience of having enzymes that have made real-world impact, and examples where design and evolution were combined, where new to nature function were generated, and even Nobel Prize was awarded. And what are these things that we cannot solve or explain at the moment. One is that not every protein is actually evolvable. There are many unreported results where proteins just never really take off in a direct evolution campaign. And then the easiest thing is just to change the protein scaffold. Sometimes there's an easy explanation that the enzymes are simply not stable enough, and that's why they kind of don't take mutations in. But often there's an intrinsic property that we don't quite understand that constitutes evolvability. Then there's an issue of the magnitude of the improvement that you can get. There are very successful evolution campaigns for demanding reactions over 100,000-fold, but they are relatively rare. And then there is this phenomenon that Dan Torvik and Nobuo Tokuriki have described as the law of diminishing returns, that you start off with a reaction that's not very well at all catalyzed by your enzyme, and you harvest the low-hanging fruit. But then to make an enzyme very efficient, you run into a roadblock, and either you run out of patience or money, or just get bored. And then these projects are normally dropped. Whereas nature, admittedly, had quite a different time scale, not the kind of days or weeks that we have available to us, or maybe even the 3 years of a PhD. But nature, of course, has centuries or millennia, really. But on that time scale, at least, the evolution campaigns look like they would always be able to be improved and in the end up at diffusion controlled rates. So we're not there. We don't have an enzyme really that has been diffusion controlled, although some designs that were evolved then go in this direction. So the question is, can we make direct evolution more predictable? And of course, the answer is no. You can't predict a parameter space that you can never explore. Sequence space is vast, just as a combinatorial consequence of the different amino acids that can be combined in many different fashions. But should we look at evolution more as a system? And the kind of intellectual concept for this is to look at proteins as products of a fitness space that is created by the sequence that are available and by the fitness. Good enzymes are up here and bad enzymes are down there. And if we only focus on individual success stories, describe this peak or that intermediate peak here over there, we might not understand what nature has to be challenged with. And that is a pathway, to find a pathway from up here, from down here to a peak over there. So should we treat evolution more as a systems biology problem that is a function of ensembles? And should we describe fitness landscapes and then look for trajectories in these fitness landscapes to find out what the best energy conserving pathway is? And should we consider the long-term prospects of protein? What will eventually lead a protein, and is it worth investing more time in screening? And to do that, we need, I think, original data sets of genotype-phenotype pairs to populate this, not just single mutants, but larger numbers of mutants. And that requires, in turn, the combination of ultra-high throughput screening, and I show a technology for this, and next-generation sequencing, where ideally we want to sequence the whole gene to look at the phenomenon of epistasis. And so the first problem is one that we try to at least make it slightly easier, using a trick that Joshua Lederberg had used in the 1950s, and then Dan Torvik and Andrew Griffiths revitalized in the 1990s, where they minimized the reaction volume from the test tube or the plate that we're all used to working with to a water and oil emulsion droplet. And these can be made as small as a cell, so you can either embed a cell in there, screen a lysate, or you can take a single gene and in vitro express it in there. Then you screen at a level of picoliter volumes, and by sheer kind of miniaturization, you can then achieve in kind of what normally fits into one test tube, you can multiply this by a factor of 10 to the 5 to 10 to the 8. And the question is just, can we make screening of these water and emulsion droplets, these new test tubes, automated and easy to use? The key bit again is that in a droplet, we have the enzyme function, we have an identifier, that's the DNA, and we need to develop, and you see 3 or 4 examples in the talk, a readout that's easy to understand and easy to read out, so that we can connect DNA sequence and the function that's brought about by it. So here's a way of making droplets at a very fast rate in microfluidic devices, oil comes from the left, oil comes from the right, and the aqueous droplets are generated at a rate of 10 to the 8 droplets per day. That's the way to do it. You build these chips yourself, and then around the kind of chip with these biopsy punches that lead to liquids going in and liquids going out and processing these droplets, you have a fast camera, pumps, and the usual kit around it. So the lab is really just a chip, but there's a kind of physical lab around it. And then microfluidic engineering has developed a number of unit operations where you can generate droplets, but then also mix them, re-inject them, make double emulsions, merge things, sort them, add reagents, incubate them for a long time. Everything you would normally do with your bare hands and an Eppendorf cube, you can do in these designs. And in the droplets, you can do anything that you can do in the test tube. You can amplify DNA, you can in vitro express, you can turn over enzyme to substrate to product with an enzyme, and you can grow cells as well. So here's a simple experiment that embodies these workflows. You make droplets, you incubate them for a long time, and then wait for fluorescent reaction product to come out. And for this, you encapsulate in a simplest embodiment E. coli that have taken a library collectively in. You put them into the droplet, they get lysed by a lysis reagent that's administered together with the oil or the aqueous phase. The cell pops open, leaves the plasmid, and allows the enzyme to turn over substrate to product. And then you look for a color, and over here you see from time to time something that is fluorescent. The camera or the laser would see the fluorescence that's emerging over here and transmits a signal to these electrodes. They change the surface of the droplet slightly, and then the good ones go in this direction, and the bad droplets that haven't fulfilled your requirement go to the other direction. And after exemplifying this assay, I want to go into a concrete example of an amine dehydrogenase, a kind of class of enzyme that Uwe has already introduced. Here, the assay is one that is done with absorbance, but otherwise it's the same. You see the droplet, you see the electrodes over here, now made of salt water rather than of soda, and the droplets are measured at this fiber optic line here, and then the good ones go in this direction, and the bad ones go over here. Now, if you then want to generate a fitness landscape, the model that you follow is that you have a cutoff, a selection threshold, and you then measure all the droplets or the clones that are beyond that selection threshold. It's really important, and some of the questions in the chat have already highlighted this, to know how different amino acid changes cooperate. And for that, you need full-length sequences. The small illumina reads might not be enough because the genes are generally longer for enzymes. And so we try to do this with nanopore sequencing to have an at-home technology for this, for long-read sequences. And the problem with nanopore is that the sequencing has a very high error rate. And so we developed a technology called UMIC-seq, where we take the genes from a selection outcome and then amplify it by transforming the library into E. coli. The E. coli makes multiple copies of the same gene. And if we introduce a UMI at the element of providing DNA to be transformed into the E. coli, we can sequence the same gene many times. And by the UMI identity, we can get an average of the sequences. And then the kind of false positives or the false mutations that are there can be removed in the consensus, and we get a quality that's as good after having sort of oversampling of about 30-fold, but as good as the Illumina sequencing. So we have full-length sequences at a very low price. We do the activity screen at kind of this very small fraction of a cent per droplet assay, and the sequence costs 1 cent per sequence. Now, if we then put them into a 2D dimensionalization, a t-SNE or a UMAP or something like this, we see that some combinations occur more frequently than others. So here is essentially something that we look onto a fitness landscapes from above, and these are the fitness peaks where something good is happening, and then there are some valleys in between. And if we now kind of analyze the sequence that are grouped next to each other, they will share a consensus sequence that's combinable. And the typical outcome would be, in this example, are 3 mutations that together make a better mutant, but we wouldn't be able to guess that if we had only looked at single or double mutations, because individually, these 3 mutations can either be positive or negative. Only when all 3 of them are together, then they have a substantial improvement. In this case, about 4-fold improvement in total turnover number. But the more impressive thing is that they're not actually residues that you would think would interact or synergize at all. They're about as far as you can imagine on this protein apart from each other. They are definitely cooperative hotspots, but this is kind of beyond any design prediction and even beyond intuition, of course, of a protein engineer. Okay, so if you do this now on a larger scale, we can describe trajectories. And here you kind of see a typical readout from these trajectories that move you through sequence space. And there are 2 examples that we look at here. One enzyme that we made more stable, but remained unevolvable, and one that we stabilized experimentally rather than by PROS that turned out to be evolvable. And so we have kind of these network graphs now that tell us about the trajectories and what makes a good or bad trajectory. So we see here that a larger sequence space is occupied, and that this profile looks very different. There are lots of little changes over here, but they are effectively unproductive because the activities don't go up. So you can change. It's not unstable. It doesn't collapse, but there's no productive element of it. And in fact, in the middle of this evolution, the bench was apparently too contaminated. So this evolution campaign stole a few mutations from here by recombination that we didn't see coming. However, this is apparently a protein that has a printout of a good evolvable catalyst. What you see here is that there are some very defined activity maxima and then highways between them that are not far apart from each other so that they can be crossed very quickly. So here's a printout or the footprint of an evolvable protein. Here's one that's not evolvable. Okay, so the next example is one where we tried to go with this to very large scales. And it's an example where we looked at the recognition of a kinase. Kinases form networks in nature where phosphorylation events are propagated. So they have to be highly specific. And there is kind of a design for that, a particular D domain that many kinases share, where they associate with the next kinase in line. And these are highly conserved in terms of their structure. And so we made a library that embodies in this D domain sequence everything that is there on the database, a relatively large library of about 10 to the 6 members, and then developed an assay that allows us in vitro to screen these phosphorylation events. So we have the gene and the substrate on the bead. We then in vitro express that makes RNA and a constitutively active kinase. This kinase then interacts with a separately supplied next kinase in line. And if successful, it will phosphorylate the target sequence on the bead that's supplied over here. Now, if we then cleave this, or try to cleave it with chymotrypsin as an assay reaction, then wherever the phosphate is, the cleavage will not happen. So successful phosphorylation prevents hydrolysis by chymotrypsin, and that means either the GFP label that's attached to this target sequence of the kinase is either still attached to the bead or not, and that allows us to sort the good catalysts from the bad ones and send it to next generation sequencing. And then we see kind of this type of pattern over here, where we have a large number of total sequences, but the good ones are in the green bin, the mediocre ones are in the yellow bin, and we can stratify our large population into useful catalyst and non-useful catalyst, and then use enrichment in each of these bins to tell what's happening. This is a typical hotspot analysis where we just kind of see what we see in the entire library in this green bin over here with high activity. So we see that some hydrophobic residues are necessary, but it becomes more interesting when we look at a pairwise enrichment where we now compare whether 2 residues are combinable in the sense that they enhance activity. This is based on the 30,000 reads in the good bin. And what you see here is that in whatever position you are at, the combination of a hydrophobic residue here and a hydrophobic residue in the second position are helpful for catalysis. Then we see high activity, and you see this as these red crosses. So hydrophobic in 2 positions helps, and the number of positions in between is 1 or 2. So we have a pattern over here, we have anchors, and we can define a motif that's apparently helpful for catalysis. Now, if we preserve this motif, then anything else goes. You see here very washed out colors, and that means for the rest of the site here, there's not much to go by. Anything goes in principle. But if we don't have this phi x phi motif, then the selection becomes much more discriminating. So what we have here is effectively a global tolerance inducer. If you have this good motif, you increase evolvability of the proteins. And when we then in a short while look at a sequence similarity network, we see effectively peaks growing into each other. Before I do this, I just want to show that we can also learn other things. Here's, for example, something that we can see when we preserve this this phi x phi motif, then there is a strong charge preference for the polar residues. So you balance the overall charge of an active site. Again, it's something that wouldn't have been obvious from a normal mutational element, but it's clearly an important molecular recognition mechanism. And after finding out that these phi x phi motifs are important, we now learn that you want in the rest of the pocket something to balance the total charge change over here. If you don't have the phi x phi motif, it looks completely different. So we learn a lot about cooperativity from these large data sets. And we see that we can go from one sort of pattern that are shown in different colors to the next one without much overlap. In the middle, you see a big gulf. And this is because some of these we introduce deletions as well. So you have to cross this deletion space. That's a difficult step. But otherwise, you can change from specificity to specificity almost seamlessly. And that's what we call this rolling hills or holy landscape model. And that provides us with connectivity. And the question that we want to answer in the long term, and after we found out now that it's a very tolerant molecular recognition scheme, can we use that to actively evolve kinase cascades? If nature has set up an evolvable system, it has used it itself, but can we also emulate this in some way? Okay, before I go to the next one, can someone tell me how many minutes I have? 5 minutes or is it less?
You have 3 minutes till the 5-minute question period. Okay,
all right. So I should be quick. Well, here's another way of improving the quality of our data. And this is where we try to do the following. We're cloning libraries, but now we would like to attach a score to each of the mutants that we find after the selection. So far, we've just put them in 3 bins. Now we want effectively every mutant to have its own score, not just to be grouped with kind of a population, but we want it to be evaluable on a mutant by mutant level. And this is how we do it. We attach UMIs to the library and then go through an error-prone procedure to make a library and then do a selection. And we can then show that the fitness score that we obtain by finding a good mutant many times in the library corresponds to activity. This is very important because that shows us that the number of sequence occurrences tells about the activity. So we want to have these fitness scores, and we actually get from these kind of numbers where we oversample a library 60-fold and then do UMIC-seq for long-read sequencing, we get fitness scores on the order of about 20,000 in a 60,000 membered libraries. And we learn something about the molecular recognition features, but we can also now see how these residues connect. And this is effectively an answer to the question that was posed in the chat before. Do we know which connections are possible? And this is a very highly complex matrix, but we see that there are some hubs that connect with anything and others that are still moderately tolerant, and other residues don't turn up at all. So we see positive epistasis. This effect of cooperativity is referred to as epistasis. And when we contrary believe, some of them are actually good when they're put together and even a bit better than their individual contributions suggested. We've put this into a machine learning model where we take these scores and mix them with an ESM model and with some physical factors. And we can then, with high probability, predict fitnesses beyond what you actually see in the library. So we get now mutants out that are, for example, require 2 base pair changes to go from one amino acid to the other based on our data set. We've also built higher order models where we work with slightly more sophisticated models where We use structural information, an elastic network model, and a decision tree that makes this automatic, and generate double mutants that that work as well. So here you see kind of what in total we achieve. We improve wild type, in this case, by a total of 16-fold. And we use both the rational design from looking at these surfaces and the AI-enabled scores. And what you can then do by virtue of our AI collaborators, Pietro Leo and Simo Mattis, is that you calculate Shapley values that tell you which effects have brought about each mutant. So for each mutant, we have a profile from this machine learning interpretation that profiles it much more than kind of years of experience with a particular protein would do. So I think we're direct evolution is still better when it's done at high throughput, but I think what we're now learning is that these large data sets contain not just good hits, but also good data. And the question is whether this is a Google moment. Will now the data we generate be more valuable than the actual hits, because we can easily go beyond that. But most importantly, we can now test these ideas with an experimental system that's relatively straightforward and compare different strategies. This is just one example. Let's see how it looks in the next case. Okay, so I thank the people who've done the work, Max Ganz, Enrique Ninzel, and Paul Zurek in particular for the IRAD project, Remke Scheles, Lawrence Lindenberg, and Maya Petek for the kinase work, and then our collaborators, Simo Mattis and Pietro Leo on the AIML, and Marco Heivern for structures. Okay, thanks very much.
Thank you. Yeah, it's really exciting what we can learn from these data that are coming out of these, big data sets. What we might learn, what we understand now, and what we may learn in the future. So, so I have a question. I'll just take a couple here from the chat. So one from Carlos Acevedo Rocha. He asks, what protein folds are not evolvable in the lab, and why is this believed to be the case? Do you think some are more or less evolvable?
Yeah, so I think the answer is it would be nice to see, for example, these network graphs. I think they hold the key to this experimentally. But again, we've only looked at 1 or 2 proteins, and are comparing them now. In the past, we've given the answer, and I think that was kind of what people would discuss, the kind of mutational load model, the kind of model that Danny Tofik and Francis Arnold in parallel reviews have advocated is this one amino acid at a time. Don't overstretch the mutational robustness of a protein. It will only be able to stomach 1 or 2 mutations. And then you have to either, restabilize it, or you have to start thinking about co-expressing a chaperone. So in the past, I think you would have said that stability is probably exhausted after a few rounds. And I think that's that probably has to be augmented by additional factors. So here, for example, we had the kind of graph network that I showed. That was 2 proteins that had same stability. One was stabilized by PROS, one was stabilized by our experimental efforts, and one evolved better than the other. It's not obvious what the reason for this is, but there are probably proteins that fit that bill, that they are perfectly fine. They're stable, robust, but they just don't want to evolve. And I think only the kind of systems data that we're generating now will be able to tell us that. I can't even guarantee whether this experiment will tell us that, but there's a chance at least.
So I guess at the crux of this is cooperativity, which you mentioned, right? So
Exactly.
Local and global cooperativity. I think this is also in the chat question. So how do you think about that? Can you yet? Or is it just experimental?
No, I think we're thinking about it. It would be really nice to know what the simple solution is. I mean, I guess the phenomenon is, just to reiterate again why this is complicated, is not because whether one residue and the other will be cooperating, but it could be a third residue or fourth or fifth or sixth, right? That destroys this cooperativity again. That's why it's a really complex phenomenon, because there's so many dimensions in it. And we are just, so we have, single, double, and triple mutants in this data set that I discussed in particular. That gives us, epistatic interactions, but we're definitely not seeing all epistatic interactions, because we are screening pragmatically, because we have to kind of set it up in this way with a relatively small library, much larger than, single mutant analysis. But there probably are surprises that we would get. And I don't know whether we understand epistasis completely, but this argument with the creationists, is always about library size, right? The creationists say that the library sizes that we study in the lab, are much larger than the ones that nature has studied. And so if we don't get anywhere in the lab, then nature can't have evolved. I'm simplifying an argument here, right? So the way out of this dilemma is that you have to say, okay, so if nature can screen with small libraries, then we should also be able to screen with small libraries. That's kind of how Dan Tovik would have put it, I think. And he and Tretznald exemplified it with some neutral drifted libraries. The general message might be that there is a combinatorial diversity out there, but that we see kind of patterns with much smaller numbers. And I have absolutely no evidence, to suggest that my 20,000 in this experiment are enough. But I want to say that we're learning something, right? And there could be a world out there, but it may just look exactly the same, that the identities of registries are different, but that there are enough pathways on this side of what we can see with, just, tens of thousands to a million or so, which we can eventually, I think, analyze and study, that we learn enough about the protein to get by. So in the kind of spirit of nature as a tinkerer, if we tinker with this kind of scale, we might be getting somewhere, but there's no guarantee it'll work.
Yeah, so it's hopeful. I think at this point my alarm went off, so I'll keep us on schedule. There are lots of questions. Uwe has been doing a fantastic job answering them, and you as well in your talk, so this is great. Thanks to the audience for giving us these stimulating questions. So we'll come back to some of them later. And at this point, I will turn it over to Ingmar, if you can share your screen.
Yes, will do. Can you see my screen?
I see it, yes, thank you.
Okay, perfect. Yes, so my name is Ingmar Schuster. I'm with Exazyme. I prepared this talk with Harry Sevi, and I will give it today. And I will talk about folding-free machine learning for protein engineering. I'm keeping it more in a tutorial style for several reasons. Number one, I think especially to many biochemists and chemists in protein engineering have seen again and again that many concepts from machine learning are new. So I think it's good to be educational and tutorial style. And number two, of course, we don't want to go into extreme detail about what exactly we do in the company, but you do get the gist of the methods that we use. So as an overview, I will give a short intro. I will look at sequence-to-function models, sequence-to-function machine learning models, which are exactly the basis of using machine learning not to predict folds in order to, like using AlphaFold in order to then use rational design, but rather directly go from the sequence to, say, catalytic speed or stability of protein binding affinity, whatsoever. I will talk shortly about how these sequence-to-function models can interact with folding and with insight about structure, be it from the lab or be it from prediction tools like AlphaFold, give some examples and then a conclusion. And by the way, let me start my own timer so that I keep an eye on the time myself. Here on this slide, I'm trying to show the differences between sequence-to-folding models like AlphaFold, and there's other examples of models that take in the sequence and give you a folding prediction, like OmegaFold, I think, is a Chinese paper, for example, versus sequence-to-function models. And the approach in sequence-to-folding models is that you take a sequence, you predict a fold, and you use structural insight then in order to engineer your protein. The advantage can be that you can train on a large data set, especially the CUSP competitions have generated data sets of sequences and corresponding folds. And you have explainability. Oftentimes, scientists go ahead, look at the predicted fold, and they can tell you, okay, if I do the following mutation, that will likely have the following effect. The disadvantage is that there is no information gain actually in predicting a fold, because the fold is more or less uniquely determined by the sequence. So from a statistical standpoint, you don't gain information if you just predict the fold. It's a different story, of course, if you do crystallography and have a measurement from the lab, because you have ground truth data then. and thus you gain information. And this is only applicable to certain protein families. Enzymes are a very nice example, but kinases, which have been mentioned before, they don't have a fixed fold because they are intrinsically disordered proteins. So there's a problem for these types of proteins. The approach in sequence to function is that we take that one data point is a sequence of a protein and the measured function like catalytic activity or stability, and that you directly predict these properties from the sequence. The advantage of this approach is that it's applicable to any protein family and function, and it's quite simple from the statistical standpoint. And the disadvantage can be that you can have explainability problems stemming from this. Now, how does such a folding-free sequence-to-function approach look? Well, basically, you start with data, ideally, of course, from microfluidics, because you get loads of it that way, but even very small data sets can work really well as long as you have low measurement noise. And the data is basically sequences and associated measured properties. And I've just generically named them Y1 and YN here. Interestingly, you don't necessarily even need to know what exactly was measured here. The statistical models don't care. As long as you have a number attached to a function, you can do predictions in principle. Then the next step is that you embed the amino acid sequences into a mathematical vector space, which is just symbolically shown here. You just make points out of these sequences. And in the next step, you fit a statistical regression model predicting, for example, catalytic speed, if that's what you've measured in the lab, directly from the amino acid sequence embedding. And the final step, which is very often forgotten in the literature on machine learning models going directly from sequence to function, is to then select promising sequences for the next step of experiments in your lab, for the next iteration in your lab. And I just want to show one example of an embedding of an amino acid sequence to a vector space. And this is the most basic example here. So here you have a comic of what's happening. You get these 4 sequences and you embed them into a mathematical vector space. And in the one-hot embedding case, this works as follows. We encode each amino acid in the sequence to a vector of zeros and ones. And the example sequence I'm using here is only 4 amino acids long. It's A, C, D, G. And you take each position in turn. And the first position, the A, you would encode as a mathematical vector by putting a 1 for representing the fact that you have an A at the first position, and for all the other possible amino acids, you put a 0. So basically, the first position is represented by 20 numbers. One of these 20 numbers is 1, representing an A, and all the others are 0 because you don't have all the other amino acids at this position. Then you go ahead to the second position, which is a C in our case. Now you put 0 for the A because it's not an A. You put a 1 for the C and zeros everywhere else. And you continue doing that. And it's really as simple as this. Now, because this is a sequence of only 4 amino acids, you have the complete representation highlighted here in light blue. It's simply this matrix of zeros and ones, which represents your amino acid sequence. And then you can go ahead and fit a simple regression model. It could be even a random forest, as Florian just showed a few minutes ago, that maps these zeros and ones to whatever property you've measured in the lab. Typically, in the company, what we do is we get data from clients who have measured in their lab whatever they are interested in. And we fit a regression model specifically to their training data. But there's another possibility, especially for properties that are of interest to many protein engineering campaigns, like solubility of the protein, stability of the protein, affinity, whatever. You can collect large data sets and train regression models once on these large data sets, and then apply this trained regression model to completely new proteins. The big advantage of this is, of course, that you don't necessarily have to measure stability, for example, anew in the lab for the new problem that you're approaching, especially in the setup where, in the commercial setup where people want to use machine learning, but they don't want to necessarily give us data about the protein that they're trying to engineer. They don't have to. So they don't have to share measurement data for this particular approach. And as I mentioned before, the step that comes after this is to select a batch of amino acid sequences for your next iteration of measurements in the wet lab. This is often overlooked. We found in our experiments that this can make a strong difference to be able to select a set of next sequences which trade off exploration and exploitation of the known fitness landscape. So Florian showed this fitness landscape typically plotted out as a hilly landscape. And you can imagine that around a peak, there's several other similar protein sequences that also have high fitness. Now, of course, you could just measure all the sequences at the peak, but the problem is you would not get a good overview of the fitness landscape. So if you would measure only around the peak, so very, very similar landscape, a very, very similar part of the sequence space, you would exploit your knowledge of the sequence space, and you would exploit your knowledge that this is a good part of the sequence space, but you do not explore enough. And here I've made a simple example where we have two variants coming out at the very top, according to a regression model that was fitted on experimental data, which look extremely similar because they share the mutation in position 227 exactly. Both of them have mutations in position 172, which are only slightly different. Now, because our regression model tells us that these three will be very high in fitness, going at it naively, you would just select the top variants as per what your regression model predicts. But in order to get a better overview, you might actually skip this third mutation, which is again in the same position, and rather go to a different position in order to better inform your next round of experiments in the lab. Then you skip a few more and go to yet another position. So this is the idea of selecting a batch to be measured in the wet lab. And I've brought a small comic about how this can be done, stolen from a paper by Gonzalez et al. The idea here is that you take a fitness landscape, plotted out here. Of course, this does not completely apply to protein sequences because the x-axis here are actually numbers, whereas for us, we have discrete changes from one amino acid to the next one. So there would actually be like a step function type of fitness landscape. So please ignore that. But on the x-axis, you can imagine these are different amino acids. And on the y-axis, you have, and this paper plots out the predicted fitness. And the idea for selecting the first sequence is super naive. You just use the sequence that was predicted to be fittest. Now, the actual innovation of this paper comes in the second, in selecting the second sequence, namely, after selecting the first sequence, you penalize the direct vicinity of the picked sequence so that you have a new fitness function, again plotted out in a solid green line, which will push the next sequence that is selected to be different from the first sequence. In this case, leading to you going to a different peak and just taking a faraway peak as the second sequence into the batch. Then you do the trick again. You have a new penalization function in blue, which you multiply on top of the penalization that you had before, giving you a newly changed fitness landscape, and you pick again from this new fitness landscape. And the idea here is super simple. It has proven to be effective in selecting a diverse batch for evaluation. This was not in the context of protein engineering, but it's completely applicable to this setting as well. Now, many biochemists we've spoken to have told us, yeah, but how can you even suggest something if you don't know the fold? Because fold is so is the central determining thing to protein function. Now, I want to give you these few points to think about. Number one, sequence, as I said earlier, for the largest part, determines folding. To a very large degree, sequence determines the 3D structure, which then in turn determines function. So of course you can predict folding from a sequence, which is what AlphaFold does, and then use the predicted fold to predict function. And while this part here is often implemented by AlphaFold or crystallography, if you go for ground truth data, this part here is often implemented by a person looking at the fold and doing and visualizing this, including the substrate maybe, in the case of an enzyme. But statistically speaking, there's nothing that speaks against going directly from sequence to predicting function. And you've seen a few approaches to that. A few other points to keep in mind are that there are some proteins that have no particular fold. For example, intrinsically disordered proteins. They have no fold, but they have function, of course. Another important point is that a predicted fold is not the fold. It's just a predicted fold. AlphaFold can be wrong, right? And this is something very important. And the third part is that dynamics are not captured by a predicted fold, of course. So this again, intrinsically disordered proteins are one example where you have several arms of a complex machine that move around in space, which is very vital to the workings of intrinsically disordered proteins, but is not captured at all by folding. But of course, you can incorporate fold information additionally. And one very simple approach is that you get predictions and even a suggested batch of sequences to measure in the wet lab in the next step. And you just let humans look at this list of suggested sequences. And they might use AlphaFold additionally, or they might use their intuition from the experiments, whatever, and filter through this list of suggestions. Another possibility is to use folding data to train sequence embeddings. The sequence embeddings I've shown you, these one-hot embeddings, they cannot be trained. They are just fixed embeddings, and there's a fixed recipe for making them. But you can think of more complex sequence embeddings that have trainable parts in them. And for these, you can use just data in order to make these embeddings as useful as possible. And for example, the ESM model that also Florian has mentioned is one example where a large data set has been used to train sequence embeddings. This was not done in the case of ESM using folding data, but rather a language modeling objective, but the idea is the same. And then, of course, you can predict a fold and then prioritize positions where you expect epistasis, because these positions all are close to the active site, for example. You can prioritize positions on the surface if you're trying to engineer for solubility, and you can prioritize amino acids that block the entry and exit channels of substrate and product, or modify it in any way. And just to show you some examples, one example which we have recently published is that of a carboxylase, where we were able to improve CO2 fixation after a previous publication by the group of Tobias Abb. They published about this carboxylase, which was a new to nature carboxylase, where they synthesized about 15,000 variants in the lab using an error-prone PCR method in 2 directed evolution rounds. So basically, they first engineered one starting point from rational insight. Then they did 2 rounds of rational design, R1 and R2, and then added on top 2 rounds of directed evolution. And you see the catalytic speed improved by a high multiplicative factor, whereas the ATP consumed per carboxylation reaction decreased. This paper was published. It is no secret, because they published it in the paper, that there were several other rounds of error-prone PCR that they tried after the DE2 round, and they were not able to further improve this enzyme. What we did then is we asked them to please sequence some of these 15,000 variants, because many of them were not sequenced, because you just don't need to in error-prone PCR to get improvement. So they sequenced 156 of the active variants from the last directed evolution round, including the best variant thus far, which they called M5. And this is the M5 variant in blue that you see here. And then we suggested, based on similar methods as you've seen before in this talk, we suggested 10 more variants. And of these 10 more variants, 80% were active, which is a stark contrast from error-prone PCR rounds, where typically 10% of the variants were active. And one of them showed a 2.8, actually, this is wrong, this is not percent, a 2.8 fold increase in activity. This is variant A here. So here you have the much more active variant. And another of the variants showed a reduced ATP consumption. And this is variant B that you see here. Unfortunately, these couldn't be combined. But it's our collaborators, Tobias Abb and Daniel Machal, were quite astounded that with these few variants, you could get these strong changes. Other examples that we have worked on in the company are engineering thermostability for industrially used food feed enzymes. Of course, we worked on catalytic speed. There was also work we did on expression and fold improvement during the protein engineering tournament, which was or is a still running competition similar to CASP that was run last year, where 4 different enzyme classes with different types of properties were in a training data set, and participants were asked to predict these properties for unseen sequences. And to conclude, folding-free approaches directly predict function of interest of any protein family. Suggestions can sometimes be hard to explain, which can also actually be good, because when people think about something, they sometimes are biased. And strength and weaknesses of these folding-free approaches can be complementary to structure-based approaches and can be combined with them. And that's it. Thanks.
Thank you very much. That was a beautiful illustration of what we can do with the machine learning. I think we'll have some specific questions now for your talk. There are many again in the chat. People have been busy answering. I think Uwe, I'll give it to Uwe to ask some specific questions. Then we'll have a general discussion.
Yeah. So Ingmar, maybe you should have a quick look into the questions,
yes,
which you can answer right now, and otherwise then we should go for some more general questions. Yeah.
Oh, okay. We have a
So
sorry.
Multitasking, right? We're all getting amazing at this.
So, well, there's one question: how much training data is needed to cover some extent of sequence space?
Well, if the question really is to cover some extent of the sequence space, then the answer is loads and loads and loads. If the question was a slightly different one, namely, how much training data do you need in order to get advantages over not using machine learning, then the answer is, well, if you have a good assay, then like low measurement noise assay, then low amounts of training data can that you feed to a machine learning system can give you a big jump in the speed of engineering enzymes.
Mm-hmm.
Like,
and there's a related question,
40.
which I also consider very interesting. Did you have the chance to model the turnover KCAT of an enzyme? If yes, how predictive were your statistical models using sequence information? So this is a property prediction, what I also mentioned in one of my slides. Can you predict something like KCAT?
You can, of course. The problem is the measurement data, really. And the fact that KCAT, like you can't, like in order to be able to use data from one enzyme and KCAT for one particular reaction and to transfer the knowledge that you gain to a even slightly different enzyme with a slightly different reaction, this is more advanced than what I showed today. I think in large part, it's a data problem and not so much a model problem. Models are there, but the data is very expensive to get.
Yeah. And then there are several questions which
discerning in reading literature, because there are many things that can be optimized. So for example, stability is probably relatively easy to optimize.
Yes.
And so if you look at some approaches like, for example, a recent PETAs from our Alpus group, that was the data set that they used to compare it was a protein beta database, PDB. So in the end, more proficient PETAs came out, but probably just because it was more stable and lasted longer. So if they evaluated it by total turnover, that's what you kind of get in such a situation. if you look at the recent TEF protease work from David Baker's group, it also looks like they might have optimized stability as well as catalysis. It's often difficult to disentangle it when you look at after a long time, right? And so the most precise system is the one by Craig Markin, developed in Polyfordice in Dan Hirschlag's lab. That's a real physical organic system where you can look at kinetics of folding and kinetics of catalysis, but it's only 1,000 mutants. And I'm not sure whether you need that level of precision. Let's see, right? But it's really difficult to evaluate. So we have to spend as a community some time on thinking about what comes out, because there are some machine learning studies with 30 mutants, and they're successful in the sense that they achieve higher rates than, you know, single digit maybe improvements, but that was comparable to the remarkable examples from today, for example. So it doesn't mean that 30 mutants are always enough, right? It may be different for every enzyme. And so just read papers carefully, because we don't know the answer, I think, at the moment. And it's very important what is actually improved. And when you took a total, it can be everything. Yeah. And just a side remark on the quality of the assays, right? So the quality of the assays in the droplets is extremely high. It looks like it's very high throughput, can't be precise, but physically they're really good. Okay. The variance comes, for example, from annoying phenotypic variation, right? So if you put a compound in that, you know, just stays in there, it's wonderfully precise, you know, less than 1% or so. The measurement is good, but when you have an expression system that has variation, that's where the error comes in. So they are very high quality systems. Dan Hirschlag and Polyfordice system is super high quality, but even these moving droplets are physically great. Microphysics, applied physics are doing a wonderful job. The biology is messy at the moment, unfortunately.
So what we all learned also from the questions is Ingmar needs a lot of data in order to have a reliable prediction. And there's a question to Florian. Would you consider to put some of your large data sets in the public domain together with a benchmark infrastructure to have an ongoing comparison on prediction methods? So of course,
we've done this.
The more data is available in the public domain, the better. So
no, it's coming out with a paper. So the kinase paper that's published already was last early 22 in Nature Communications. The first author is called Scheele. And that data set is available, and we're doing kind of generative modeling on that together with Christine Orengo. And that will also come out. So that's a data set that I think is amenable to prediction. And it would be great if you tried your hand on this with a different algorithm or with the same one, and let's compare what the what the outcomes are. So we can predict, I think, our own data set now. And we're still doing experiments to show they can predict beyond our own data set. But the data are published already as Excel sheets.
Ingmar, do you also look into the F&A question and answer?
Yes.
Yeah, so is there maybe one of the 5 or 6 remaining questions you directly want to answer?
Yes, maybe the one that comes out at the very top for me. How do you separate sequences into training validation test sets? I think the question is informed by the CASP competition. The CASP competition focused very much on out-of-domain predictions. So basically, the idea was you had a structure that you have to resolve that was not resolved before, and you were and teams were urged to not use any data that is close to what you're trying to predict. This is a particular challenging problem, this out-of-domain prediction. It's not necessary really to do this if you are in a setting similar to directed evolution. Of course, it's the harder problem that you can face. And one way to then split the dataset into training validation tests is to do clustering, for example. And based on clusters, so you basically take clusters of similar sequences, and all of them go into the training data set, and clusters of other sequences that share little with the stuff that's in your training data and put them in the test data set. So that is one idea how you can do a split of a data set.
Okay.
If you want out of domain prediction, of course.
Here's another short question. How Exazyme addresses epistasis?
Epistasis.
Epistasis.
For epistasis, so one answer can be a lot of data, but to slightly correct you, Uwe, we don't necessarily need a lot of data. If you go for one hot encoding, there's no way around a lot of data for epistasis. But you can have these trainable embeddings that I was talking about, and these can often capture epistasis information already. And one way people have done this is using so-called language models. And the big advantage of language models is that there's loads and loads and loads of data. The idea simply is you have, for example, the start of a protein sequence, and you are trying to predict the last 20 amino acids of this protein sequence. And this is very cheap data to get from UniProt, for example. What people have shown is that just solving this task already teaches you about epistasis and teaches you about fold. And so this is another possibility to deal with epistasis. Yeah.
Mm-hmm. Okay.
I might add a general question, actually.
Yeah. Mm-hmm.
So it's I think what I'm hearing sort of from thinking about this field is that, you know, amazing things are being achieved. And now we have these new tools of machine learning and high throughput and getting all these data that we can improve our machine learning with, combined with rational. It feels like, you know, in the precise outcomes, we still have big challenges, right, to make really good things. We can, practically speaking, improve many proteins in many ways. But in terms of precision and the big picture, we still have a long way to go. And I guess the sort of general question that I have is, and it came up in the questions earlier, is how far along are we in using AI approaches and machine learning approaches to effectively engineer? How far are we?
There's still loads to do, I would say. There's quite a few approaches that have not been developed to the end. One of the reasons being data availability. Another reason being different schools of thought, I think. So whenever I talk to people that have a biochemistry background, they think a lot about structure. Speaking to some people that have a strictly machine learning background, they shove that all away. And I think some approaches can be much more simple if you don't have to think about structure. If you only think about statistics, I think you can go very far sometimes with seemingly simple approaches.
Would you agree, Uwe and Florian?
Yes.
I would just put a different spin on it, because I think there might be different stories for every mutant that we predict. And I find my collaborations with the machine learning people really helpful in that respect, because they calculate these shapely values and they can describe, you know, what was important for predicting this particular mutant. And I think what will emerge eventually is that some things can be predicted well for one particular fold. And so then you know what can be expected of a machine learning approach. That's much. It's answering the question in some way. How much can you expect? But how much can you expect in total is a big question, and it's kind of parceling it out. Can we expect for this protein, you know, this effect? According to previous predictions, the shapely values told us that, you know, the ESM is helpful for doing this, or the parameterization is helpful, or the machine, or the kind of experimental data set. And so I think that's kind of something that will guide us in which direction the machine learning is best used, and when it's kind of naive to hope for, redemption, and when it's actually a matter of, muscling in and just waiting a bit longer or doing more experiments.
Mm-hmm.
Mhm.
Uwe?
We still have so many questions, even if we answered already many.
Well, yeah, so what,
you-
yeah, I know you guys have actually been fantastic at answering the questions. And unfortunately, they do disappear at the end of the webinar. We have a few more minutes. So I feel like if there's any sort of big picture things that you'd like to return to, or really important things in this field, I'd like to take a few moments to let each of you think about what do you think is next? You've already told us a lot about that. What's the key challenges? Addressable, grabbable challenges?
Yeah. So for myself, the answer is look into our review published in November last year, because we thought a lot about what comes next. Yeah? So I think because it was published just a few months ago, this is the view of us 6 authors, what we consider the emerging technologies, or where we have bottlenecks, and what comes next. Yeah.
Great. Yeah.
Yeah.
You won't be surprised to hear me saying this, but I think it's becoming back to mechanism, right? I think we can be much more entrepreneurial in traversing sequence space, and so mechanistic stories that sometimes are, made impossible by insufficient stability or by something like this. We can shape our strategies more according to these. So I think after this kind of interlude, of big data, I think we'll come back and converge again in mechanistic enzymology. That has become a bit, I've kind of talked it down in my talk, because I said if you only characterize one mutant, you don't learn that much. I think there's a mechanistic enzymology that can take kind of the entirety of a data set and identify mechanistic stories that then become traceable again. And so many things, I think, for proteins are not traceable, because proteins are intrinsically difficult to study. We know how the funding system looks at complex projects sometimes without a direct application. But I hope that the machine learning and the large data set interpretation will help us to focus our mechanistic efforts again. I think that'd be really important, because I want to understand fundamental phenomena in addition to saving the world, I think.
And I think we need all 3 approaches, right? We need the sort of fundamental, the practical, the things we don't understand but can use new tools to address with machine learning. We need to integrate them all, right? I think that's what's kind of come.
And
Exactly.
I fully agree to what
Accelerated by all these other techniques that are there, right? The high throughput, the large data, and so on. They facilitate studies that otherwise would have been completely unfeasible, I think. And that's why I think the mechanistic area kind of went to sleep a bit, but I hope it's going to wake up again.
Yeah. So I agree, because what we learned from mechanistic studies is also enzyme promiscuity. Many promiscuous activities were discovered because people looked in detail into the mechanism and came up with an idea how to change the protein that it catalyzes a new type of reaction, natural reaction, and all the new to nature chemistry, or what is published as new to nature chemistry. People had to look into mechanism, transition states, think about how can we implement a new type of reaction in this protein scaffold with a few changes. So, and then they could do exciting new chemistry. So I think this definitely is a key building block to bring biocatalysis in a broad sense further, but also to improve our understanding of enzymology. Clearly important. Yeah.
And Ingmar, I see your hand.
Yes. I think really something that we've all hinted at, collecting data that is transferable is surely one way to do less experiments in the future, and thus increase the cases in which we can use like enzymes, for example, if we are trying to replace standard catalysts like non-organic catalysts, and also increase the use of biologics in pharma. The other thing that I think will be very important in the future is to really get things that have been developed in the lab and productize them. And this is a very industry viewpoint, of course, but it's completely connected to transferability of data. As soon as you can use cookie cutter methods again and again and again, you have the advantages of scale, basically. So this, I think, will be very important, that we develop software that is like CAD tools for architects, for example, right? Every architect uses the same type of software tools, and they can be applied to any type of building. Just like this, protein engineers will be able in the future to use extremely similar and even user-friendly software that people have optimized for this, rather than they have optimized for writing a paper, which of course is the currency in academia, and it's extremely important. But the next step brings us scalability.
Well, I see. Yes, this is inspiring to me. I'm really excited at all of the things that you've covered here. We're at our time slot. We still have actually a lot of people and a lot of questions. This will be recorded and available on YouTube afterwards as well. But the questions do disappear afterwards. So if you would like to go a little bit longer, we can, and take a few more questions if there's anything you would really like to still address. I'll leave it to Uwe. Uwe, you
Yeah,
are.
I'm looking at some questions are really specific to Ingmar or Florian. Maybe they can answer them in the chat. Someone asked, I have a question about data reliability, which are available in the public domain about activity, different lab generated data and different assay methods. Yeah. This is the problem we always have. Yeah. When you try to write a review about a specific subject and you read 100 papers with 100 tables of data, they report differently. Some write units per milligram, some write the KCAT values, some write percent conversion and whatever. And then how can I compare this? And even if all of them use purified enzyme and not just cell lysate, it's impossible. So there is always a huge error in the data reported. And I already answered another question. Try to make your own data, look into literature for other compounds or whatever, or related enzymes, but then have one person working on this and produce data with your methods, with your gas chromatography, HPLC, photometer, or whatever, because then you have reliable data on your own, and then use this as a basis for the machine learning data sets. The major problem is that groups are working on that. There's an initiative in Germany also funded by the German Research Foundation to have the reporting of data for all catalysis disciplines in a machine readable and similar format, so that in the future we hopefully just have a website or so where this is implemented to facilitate data management and at the same time to facilitate machine learning. But this also requires that all scientists who work in the field take their time to convert their data into this sort of format. For protein sequences, it's easy. Yeah, that's why we find the protein sequences and we have all the data in the PDB file. But for the activity of the enzymes, the substrate, the products, and this is a major challenge, yeah, and source of error as well.
Yeah, so we're still at the stage of needing to organize better on this. You know, many data sets have become larger, but these ones are still a challenge.
I would also be, I mean, so there's one way, the Stranda way is to kind of just get data fully described, right? That's, I think, minimum standard of publication. That's a really good effort. And I think they're also thinking about preparing it for AIML. But I think they will never get the kind of numbers that you get in terms of diversity, you know, from a single lab high throughput experiment like the droplets, for example. So there, it's a slightly different thing. So I think it's so the sequences are in the European Nucleotide Archive, for example. That's relatively easy, right? And there it's only important that the experiment is kind of consistent. So you get scoring data for that. But the philosophical is what I said before, that, an assay can be internally consistent. But if you look at turnover after a certain time, stability, can be insufficient for some mutants, so that catalytic activity drops down, not because it's not catalytically active, but because it denatures effectively. And so a kind of a description, of when that's the case, for a particular assay, that would be important. Then someone would know, who could use these data, that my assay encompasses not just, a factor of that is corresponding to initial rates, but late turnovers, or a temperature exposure for a certain time, and so on. So that two different things of describing. So the Strender will not kind of tell you what how a high throughput experiment should be described. So there's a different kind of set of requirements.
And what is also missing is that you make 100, or you find 100 variants of a protein of interest containing certain mutations with certain effects, but you don't upload the sequences into the PDB or in the protein sequence database. There is the original wild type sequence. You start from this, but the outcome of your study usually doesn't end up as information in the protein sequence database. And even if it would be there, it's not connected to the properties you have found. So you need both. Scientists have to report the mutations not only in a table, but the entire sequence should go in the public domain, so that you can learn from that. And at best, of course, connected to the property.
I think this is conceivable. This is conceivable. Yeah, it's okay. So I think this is the challenge for everyone, right?
Yeah.
I think we've landed at a good place here. I'd like to thank you all again. This was really a fantastic webinar. I'm really excited. I'm going to think more about how we can use this too, and I'm sure many, many in our audience will as well. We still have lots here, actually, so we could keep going, but another time. Thank you again, Uwe, Florian, Ingmar. This was great.
Thank you. And we have to thank all of the people who participated and listened to our talks, and also wrote so many questions. So I found this also very stimulating. Thanks a lot, and have a nice evening for all the Europeans. Yeah.
Yes, everywhere around the world.
Okay.
Have a great day.
Thanks, everybody. Bye.
Bye-bye.
Bye-bye.